Hello everyone, this is MEENU AGRAWAL continuing with the same topic. And I told you that the plasma membrane is very important structure, isn't it? It's giving, no, it's separating the cell from the external environment. Now we have discussed in the previous class about diffusion, osmosis, which is also a type of diffusion. Now we will deal with passive transport, active bulk and bulk transport. Okay, let's coming back to the topic, and we have seen, and just I will review that thing that the membrane has got, it has got the lipids, and you know that the non-polar substances can easily diffuse through it. But and this kind of diffusion, where non-polar small molecules diffuse, small molecules diffuse very easily through the membrane. Okay, and this kind of diffusion is called as simple diffusion. And you know that diffusion is affected by the concentration. More the concentration, the more the diffusion will be, more the surface area of the membrane, more the diffusion will be. We increase the temperature, diffusion will become faster. So all these things, they affect your what? Now diffusion, the thickness of the membrane, all these things affect your diffusion. Okay, your thickness of membrane. Okay, so all this affects the diffusion. Now we also know that there are non-polar molecules. Okay, non-polar, we have talked about polar molecules. Polar molecules, and these molecules, they can't move through the lipid layer, therefore they need a protein. And since they are being facilitated by a protein, and then something is helping, therefore it is known as facilitated diffusion. And remember, it is happening along the concentration gradient. Remember, and this concentration, I have told you, it acts as a potential energy itself. It acts as a source of energy to help the molecules to move. Okay, so this is your facilitated diffusion, which happens with the help of your carriers and all. So let's now move on. As you can see here, this is your non-polar molecule, and this is what outside of the cell, and it's written extracellular fluid, and this is the cytoplasm, so it is the inner part of the cell. So you can see clearly how the molecules are, with passage of time, they are diffusing inside the membrane. Okay, with the help of your these carriers. Okay, you can see here that this is your what carrier molecule is being shown here, isn't it? This is your carrier molecule, and this is basically your glucose transporter. We call it as glucose T, Galt 1 transporter. Okay, so you can see here that there is a this glut transporter is there, the glucose transporter is there, which is transporting. Now as this glucose molecule binds to is transported, it brings about what? Conformational change in this molecule. The shape of the molecule changes. As you can see, from here to here it changes, and it releases it into the inner side. This is the outside part, outside of the cell, and this is the cytosolic part of the cell, as you can see here. Okay, so this is how your these now this is facilitated. This diffusion is being facilitated with the help of, facilitated means it is happening with somebody's help. So it is happening with the help of these carrier proteins. There are also channels regarding this, as I can show you here. You can see here that this is a channel. Okay, but these channel, they work in response to a stimulus. So there has to be some stimulus. Okay, so as you keep on reading nervous system, hormone action, you will come up to know about various kinds of stimulus that acts upon, and that is causing, see, you can see here that here is the gate, you can see this gate is opening. Okay, so when they get the stimulus, what happening, suppose there is a stimulus, could be your any molecule which is binding to this and it is causing the gate to be opened, or it could be difference in potential, no, of the membrane. So you will, as you will read, you will come to know about it. Therefore, what happens, any stimulus it could be which is causing this gate to open. So you can see here the concentration of potassium is high, so it is moving from here to here. It is along the concentration, I am again repeating the term, along the concentration gradient. Okay, so this is why they are able to move, because this potential energy is being utilized, remember, therefore they are able to move. So it's a energy that they are possessing, so therefore they don't need any extra energy. But for active transport to happen. Okay, now coming to active transport, as I told you, active transport, it requires what? It requires an energy source. It requires an energy source. Why energy is required? Because here the movement is against the concentration gradient. That is, I told you that there is a gradient, you know. Now automatically things can move from higher to lower concentration, like things if you leave in water also, na, downstream it can move very easily, but to move things upstream you require source of energy. You require what? You need to give what? Energy in this direction if you have to move things. So remember, for active transport what is required? Energy is required, and this energy is provided by the cells by ATP. You know that the ATP is the energy currency of the cell. Now if we talk about ATPase pump, so in case of your ATPase pump, what is happening? You can see, suppose I make, this is your membrane. Okay, this is your membrane. Okay, now here the pump is, okay, just let me, okay, let me get some space so that I can make it better. Now suppose this is your membrane. Okay, so the membrane I'm showing here, and this is the inside part of the cell, and this is the outside part of the cell. Now suppose inside the part of the cell, what is happening? This is your pump. I'm making a pump for you first. Suppose this is your pump, and it has got two regions to bind with sodium, and it has potassium, and it has got three regions to bind with sodium. Okay, so this is your pump, and what is happening here? You know that the potassium molecule, you know, K plus inside the cell is more. Okay, fine, and your outside is your sodium. Now since they are moving against the concentration gradient, means here what is there? Here you can say the potassium is less, and here you can say the sodium is less. Okay, so more and more sodium is outside, so sodium is less here. Okay, now this pump, what it does, your sodium binds to these three molecules of sodium will bind here, and two molecules of potassium will bind here. Okay, and on binding of an ATP molecule, ATP, okay, which will cause what? Hydrolysis. This ATP will hydrolyze to give you what? ADP plus Pi, and in that process it will give you a energy. It will give you what? High amount of, there will be a burst of energy. Okay, so there will be energy given out by this process, and when ATP binds, there is a conformational change in the structure of the molecule. And what it does, what it will do, this molecule, what it will do, you can see when this this will bind, then it will cause what? Suppose I have made, it will cause the change, it will cause, this molecule will cause now your 3 sodium molecules to move out of the cell, and it will cause what? Your 2 potassium molecules will move inside your cell. Okay, so that's how they are now acting. So very briefly I have narrated here that the 3 sodium molecules bind to your transport protein, then phosphate group is transferred from ATP to the transporter protein. This phosphorylation causes the carrier protein to change their shape, and releasing 3 sodium outside, 2 potassium binds to transport. Okay, so this is a case of your co-transport, where 2 things are being transported together but in opposite directions, which is also called as antipode. Always your sodium will be, this ATP will be required. There is an indirect way also this happens. See, I told you that potential energy is a very high energy source. Even your turbines and all in dams and all, what happens happening with a high, at a high the water is there and it drops on the turbines and it rotates and produces the energy. You can see that due to position also the energy is there, a huge amount of energy is there. Look at this case. You can see here what is happening here. This is the case of your sodium potassium pump, which I have just now shown to you people. This is the case of your sodium potassium pump. This is what is happening. See, the potassium is moving inside the cell. Okay, so if I have to write, this is what? This is the outside part of the cell. This is the inside part of the cell. Okay, so what is happening? You can see here that the potassium is moving inside. Okay, potassium is moving inside and ATP is being utilized in this process. And you can see here, now sodium is moving out because it is an ATPase pump. Okay, so sodium is moving out. Now the sodium is, due to this pump, the sodium is outside more. Due to the sodium potassium pump, what is happening? It is creating sodium outside. The gradient of sodium is more, which is acting as a potential energy here. In the case where you can see the glucose is where? More inside the cell. The glucose is more, but still it is able to move inside the cell along with the sodium. Along with the sodium, it is able to move because of the potential force of the sodium, and indirectly ATP was being utilized. Okay, so these are the 2 ways, and you can see there are even a co-transporters we talk about. Okay, so this is a case of a co-transporter. There are different types of transport. There could be uniport, that is only a single now uniport is there, where what is happening, this molecule is there, this is basically now pumps. They are basically your pumps, and remember that they use ATP. Okay, like in case of your proton pump, you will read about them in case when you will read about respiration and photosynthesis. So when this proton pump is pumping hydrogen, okay, so from the here hydrogen will be more, okay, and here hydrogen will be less, so it is pumping. So this is uniport, only a single molecule. But this glucose and sodium, this is the case of what? This is the case of symport, where the 2 molecules are transported towards the same direction. Both glucose and sodium are being transported inside the cell. And you can look at this case, which is a case of what? Antipode, where different substances are being transported oppositely, opposite directions में transport हो रहे हैं. You can see here, आप यहाँ पे देख सकते हैं कि पोटेशियम inside जा रहा है और सोडियम बाहर जा रहा है. So this is your antipode example. You can give example in case of antipode, your sodium potassium pump. Sympod, you can give example of your glucose and sodium being transported together, and unipod is your what? Your proton pump. Okay, so these are the different pumps, and they are all, remember, an example of active transport. Okay, now the question arises ki how? Now I'm asking you another question ki how do? Now these are about small molecules. Okay, now how do large molecules move inside the cell? Like if I talk about larger polysaccharides, larger proteins, then how do they move inside the cell? If we have to talk about them, how will they enter these biological membranes? And अगर ये through these pumps and all they were able to move, then तो भैया what they, even your hemoglobin molecule can move out of the membrane, isn't it? But see, look, this membrane is such a dynamic structure that it is separating, it is keeping the internal milieu quite homeostatic. Okay, so it is maintaining the internal equilibrium, you can say. So it is maintaining. So it is how it is doing. How is it doing? Then how will uh, your these larger substances will move in or out of it? This is a question. Here comes the process of your bulk transport. And you must have heard about the process which is called as via the vesicles movement. It happens. That is your by endocytosis. Okay, or exocytosis. Okay, so this is now fortunately now you can think of consequences, and this is what the first thing that happened. This membrane now at the time of evolution was able to maintain its internal milieu, and it is because of this bulk transport substances are able to move out of and inside the cell. That is exocytosis. Something if cell wants to move. Now exocytosis, that is secretion. If something cell wants to secrete and wants to move out of the cell, it does exocytosis. And if it wants to do, it can do endocytosis. Okay, now this endocytosis is further divided into your what? If you just look at first exocytosis and endocytosis, this is your what? Endocytosis, and this is your exo. As you can look here, that this now here this molecule now it is being taken. Here is a small invagination. The first thing that happens is that the first process that happens is the invagination. Okay, a small pocket is formed around that the material. Okay, that then material gets now inside that pocket. Okay, and then it completely comes into the interior of the cell, and then lysosomes bind and digestion happens and further things happen. So this is the process of your endocytosis. So we can say it moves in this direction. Okay, so it is engulfed. Okay, so this is your outside part of the cell, and this is the inside part of the cell. Okay, now if you look at the exocytosis, I hope you are clear with the outside part of the cell. This is the outside part of the cell, and this is the inside part of the cell. Okay, now here you can see the any substance which is being formed, it gets wrapped up in your vesicles, isn't it? आप भी किसी को कुछ देते तो किसी चीज में देते हैं, isn't it? Similarly, the cells also wrap these substances inside these bags, which we call them as vesicles. Okay, and these vesicles, they fuse with your, ultimately they, like here you can see it is fusing with the membrane. Here it is fusing with the membrane, and then it just releases the contents outside. So its movement is from here like this. It happens, you can see here. Now it is being secreted outside the cell. Now if you look at endocytosis, it is of two types. What are they? They are phagocytosis, like it is showing here any bulk material is being taken, and another is pinocytosis, which is where the liquid substances are taken in by the cell. We also call it as cell drinking. Now, like the cell drinks something in this case. Okay. So this is an example of your endocytosis and exocytosis, which basically happens inside the cell. Okay. So with this I will like to end, and we our transport, active transport, passive transport and bulk transport is finished, which is very important, and in the next video we will start with the organelles. So thank you for watching. God bless you. Bye bye.